Now look at this.
What's up about penguins? Today we're going to do topic 3.6 on cellular respiration. So there's 3 steps to cellular respiration. You're going to have glycolysis, Krebs cycle, and then oxidative phosphorylation. So I'm going to try to keep this as simple as possible because all you really need to know is the big picture, right? What goes in, what comes out, where does it take place, why is it important? So first, let's look at glycolysis. Glycolysis is going to start with glucose, and it's going to end up with 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. The NADH molecules, and I'm going to tell you a lie that I tell my students, that H stands for holding. So if you see you have an NADH molecule, it means that it's holding an electron. So NADH is then going to hold those electrons and take them to the electron transport chain. The pyruvate is going to move on into the Krebs cycle, and then the ATP is, of course, substrate level phosphorylation. Yay! So after glycolysis, if oxygen is not present, the reaction is going to move into fermentation. So fermentation is what's going to happen to regenerate that NAD plus that's needed for glycolysis. So if you ever talk about fermentation, it's only going to produce 2 molecules of ATP, and then it's going to go through fermentation to regenerate that NAD plus. If oxygen is present, it's going to go through the Krebs cycle, and then it's going to go through oxidative phosphorylation, which includes the electron transport chain. So before we can move into the Krebs cycle, we're going to go through something called pyruvate oxidation. This is where we're going to oxidize pyruvate. If you remember, oxidation means we're going to lose electrons. So when we go through pyruvate oxidation, we're going to take off electrons, and so they're going to be held in NADH. That's going to move to electron transport chain. We're going to release 1 carbon molecule from the pyruvate, which will make the acetyl group. And then we'll add a coenzyme A, so that's now why we have acetyl-CoA. Acetyl-CoA will then enter the Krebs cycle, and through 1 turn of the Krebs cycle, we're going to have 3 molecules of NADH, 1 ATP by substrate level phosphorylation, 1 molecule of FADH2, which is just like NADH, just not as strong, and we're going to release 2 molecules of carbon dioxide. We're going to have to go through this Krebs cycle twice in order to break down 1 glucose molecule. Then we move into the oxidative phosphorylation. Oxidative phosphorylation is where the NADH and the FADH2 is then going to release its electrons, so it gets reduced. So it's going to drop them off at these things called cytochromes. Cytochromes are then going to allow the electron to drop in energy. As it moves down in energy, it's going to pump protons against their concentration gradient. So they're going to pump those protons into the IM space, or the intermembrane space. Again, we look at the fact that if you increase the concentration of the hydronium, you're going to decrease the pH. So the IM space has a very low pH. The second part of oxidative phosphorylation is something called chemiosmosis. Chemiosmosis is where we're going to use that proton gradient. We're going to run it through ATP synthase so that it's going to be able to synthesize ATP. So clearly, I'm not going to finish this standard. So stay tuned for part 2. Remember, API is just a test. Bye.